This is the Science of Sci-Fi lecture, and this lecture is on the Half-Elven genetics in Middle-earth. So the Half-Elves were composed of two races, and that's elves and men. And the first race I wanted to discuss were the elves. So elves are the fairest beings in Middle-earth. They're known for their great abilities in battle, but not just their great abilities in battle. They're also known for their excellence in things like art, music, language. They're generally pretty tall and pale-skinned and have great agility, sight, and hearing, which really helps them be great warriors. And I've just included a few pictures of some famous elves like Galadriel, the female elf up at the top there, Thranduil in the bottom left, and then the elf that you guys can probably recognize the best is Legolas, and he's played by the very handsome Orlando Bloom in the movies, as you can see. And the second race that's important for this presentation is men. So men tend to be tall, just like the elves, but usually they're not as pale-skinned, and they also lack the incredible senses that the elves had, so they couldn't see for miles or hear everything that the elves could hear. But they also excelled in things like battle and the arts, so they were very good warriors and very good artists. And here's just a few pictures of some famous men in Middle-earth, like Bard the Bowman there on the left, and Aragorn there in the middle. So the Valar. So the Valar are sort of like the gods of Middle-earth, and when the world was created, they helped shape it. So they lived in a place called Valinor, which was sort of like a heaven on earth, which was across the sea to the west of Middle-earth. And the Valar that we're most concerned with is Mandos, and he's known as the Doomsman of the Valar. You can kind of think of him as the Grim Reaper. So the big difference between elves and men is obviously that elves are immortal, which means they can't die from things like disease or old age. They can only die from being killed in a battle, or they can die from grief. And once they die, they can either be reincarnated by Mandos or stay in his halls until the world ends. Men, on the other hand, are mortal. They can die from things like old age, disease, dying in a battle, etc. They can die from anything that, we can die from in real life. But when they die, their spirits get to leave the world and cannot be reincarnated. So that's the main difference between these two races, and that's going to play a big role in our discussion about the genetics of half-elven people in Middle-earth. So who were the half-elves? So the half-elves, the very first half-elves were a male called Arendil and a female called Elwing. And they were both products of a marriage between a man and a female elf, which made them half-elves. And they went on to be married themselves and have children of their own. And this line of descendants are also considered half-elves, and these beings are given the choice. So remember the fates I just explained on the last slide. So elves get to live forever, and men, they get to die and leave Earth. That's their individual fates. But these beings, the half-elves, are given the choice. They can either choose the fate of the elves and be immortal, or they can accept the fate of men and be able to leave the world through death. And just reading the names on the right here. So the half-elves consist of Arendil, Elwing, and their two sons, Elrond and Elros. And then Elrond had three children. His two sons there, Aladdin and Elrohir, and then his daughter, Arwen, which you might recognize from the movies if you've seen them. And you might think that one does not simply choose to live forever, like Boromir here, but in fact, they do choose to live forever. So, since Mandos is the doomsman of the Valar, once the half-elves make their choice, if he was going to decide, okay, since you chose the fate of the elves, you get to live forever, or since you chose the fate of men, you're going to have to die from something, whether it be old age or disease, I believe that he would have to perform some genetic engineering. And to me, the thing that would make most sense and would be easiest for Mandos to perform this sort of divine engineering would be divine CRISPR-Cas9, and that's how he would edit the DNA of these half-elves once they made their choice to either be counted amongst the elves and be immortal, or be counted amongst the men and be mortal and be able to die and leave the world. So what is CRISPR-Cas9? So CRISPR stands for Clustered Regular Interspersed Small Palindromic Repeats, and basically Cas stands for CRISPR-associated proteins. And this method for genetic engineering was adapted from a naturally occurring genome editing system that's present in bacteria, and it consists of two main components. The Cas9 protein, which cuts the DNA and the guide RNA that recognizes the sequence of DNA that we want to edit. So how it works in bacteria is when the virus enters the bacteria, Cas proteins cut a segment of that viral DNA. And once that viral DNA is cut by the Cas protein, it can be integrated into what's called the bacterium's CRISPR region. So those integrated DNA segments, once they're integrated into the CRISPR region, can be copied into short pieces of RNA. And these short pieces of RNA are capable of binding to the Cas9 protein. And that binding forms a complex that consists of that RNA and the Cas9 protein. And that complex itself I like to think of it as a guard dog. So when you think of a guard dog, if he finds an invader, that guard dog is going to rip that person to pieces. And this complex sort of does the same thing as a guard dog does. If a piece of viral DNA that's complementary to that RNA attached to that Cas9 protein is recognized or penetrates the bacteria cell, the complex will bind to it, and the activity of the Cas9 protein, it can cleave that DNA, it can cleave both strands of that DNA and destroy it. So in the lab, scientists developed a guide RNA to match the gene that they want to edit and attach it to a Cas9 protein. And these guide RNAs sort of direct the Cas9 to the target and the protein snips that DNA and creates a double-stranded break. So if you're in a lab and there's a particular gene that you want to target, you can synthesize a specific RNA that will recognize that gene sequence that's complementary to it. And if you have Cas9 also attached to that RNA, once that RNA recognizes that gene, you can actually sort of cut genes out. And once you cut them out, you can either replace them or you can just leave it cut out and sort of like delete that gene. So you can either delete genes or you can add genes in with CRISPR-Cas9. So in the lab, once that DNA is broken by the Cas9 protein, the cell is going to try to repair it through the cell's natural repair mechanisms. So one option is non-homologous end joining. So in non-homologous end joining, proteins called Ku proteins bind at the broken ends once that Cas9 protein cleaves the DNA, and it recruits DNA-dependent protein kinases. And then once those protein kinases are in place at the ends of the DNA ligase then binds to the complex and rejoins the ends. So it's just sort of like the cell goes into shock and it says DNA being cut in half is not a good thing, so we're just going to join those two ends together as best we can. So when you allow that to happen, you can accomplish a gene deletion. Whatever gene was complementary to that guide RNA you inserted, you've essentially cut it out and just deleted it. So now the products of that gene can no longer be synthesized by the cell. Another method for repair that the cell could use is homology-directed repair. So instead of deleting genes like non-homologous end joining, in the lab, this method is used for gene insertion. So once you insert the guide RNA attached to that Cas9 protein, researchers can also insert a template DNA into the cell. And this template DNA is flanked on both sides. So on both sides of the template DNA that you insert into the bacteria cell, there are regions of DNA that are homologous to the end of the cleaved DNA. And what this does is it allows the template to be integrated into the cut site. So essentially, you've inserted the guide RNA in the Cas9 protein, and you've also inserted a gene that's able to sort of fit in to the place of the gene that you cut out like a puzzle piece, and it basically gets attached at the site where the cleavage occurred. So it's important that when you're inserting genes, they're inserted to areas called safe harbor sites. And these are essentially areas of the cell where you can insert DNA or delete DNA, and it basically won't be fatal to the cell. So essentially, that's how Cas9 protein, that's how CRISPR-Cas9 allows you in the lab to either delete or insert genes. You can either allow the gene to be deleted, and then the DNA repair itself through non-homologous end joining, or once you insert the guide RNA with the Cas9 protein, you can also insert a donor DNA, and that donor DNA can be incorporated into the DNA strand through homology-directed repair. So now that we know how Mandos could delete or insert genes depending on whether a half-elf chose the fate of elves or men, we need to start thinking about what type of genes or what certain genes would he want to insert or delete depending on the choice you made. So one gene that he would likely either insert or delete is called the SIRT6 gene, and this is known as the longevity gene. So SIRT6 is a very interesting gene, and it has been found to play an important role in organizing proteins and recruiting enzymes that repair damaged DNA. And as we all know, more efficient DNA repair is essential for a longer lifespan because as you live, you just DNA damage is naturally occurring. You can't stop it. But if you have very effective DNA repair mechanisms, um, your lifespan can be prolonged. Another group of genes that Mandos might play around with, either delete or insert, um, are genes involved in telomerase production. So, telomerase is an enzyme that is responsible for telomere replication. And telomeres, as we all learn in Gem Bio, are the protective caps at the end of the DNA that protect the ends of each chromosome from being damaged. And as replication cycles occur, telomeres get shorter and shorter and shorter just due to the nature of DNA replication. And if they get too short, our cells age and stop function properly. Once those telomeres would stop functioning, we'd see aging of the cells. So Mandos could also possibly play around with any genes involved in telomerase production because that would ultimately impact the telomere maintenance. And lastly, another interesting gene that Mandos could play around with in these instances would be the P66SHC gene, which encodes for a protein that regulates levels of reactive oxygen species. And reactive oxygen species, for anyone that's taken biochem, are cytotoxic, so they actually can result in cell death. And studies have shown that removing this P66SHC gene can increase the lifespan of mice by 30%. So these three genes, or these three groups of genes, could be genes that Mandos would want to insert or delete depending on the choice of the half-elf, whether that half-elf chose to be counted amongst elves or men. So those who chose the fate of elves in that list that I gave you of the half-elves include Arendil and his wife Elwing, and then Elrond shown there at the bottom. So for these individuals, Mandos will likely insert the SIRT6 gene using the CRISPR Cas9, is what I hypothesize, to boost their DNA repair abilities, because if he's able to boost their DNA repair abilities, they won't be as susceptible to aging due to DNA damage. And I also hypothesize that he will insert genes that encode for telomerase, because if he inserts genes that encode for telomerase, the telomeres would be maintained better, so we wouldn't see the shortening of the telomeres, those protective caps at the end of chromosomes. So we'd also avoid damage to DNA in that regard. And lastly, I believe he would delete most, or if not all, copies of the P66XHC gene that is known to produce reactive oxygen species, because if he can limit the amount of reactive oxygen species produced in the cells, he would prolong cell life. So by inserting that longevity gene that would assist in DNA repair mechanisms, inserting genes that encode for telomerase to keep those telomeres at a good length at the end of the chromosomes, and knocking out any genes, any P66SHC genes that could produce cytotoxic reactive oxygen species, Mandos could utilize CRISPR-Cas9 to prolong life for who knows how long for these half-elves who chose the fate of elves. But surprisingly, hard to believe, but there's actually half-elves that choose to be counted amongst men. And this includes that above picture is a picture of Elros, who was Elrond's brother, or Rendil and Elwing's son, and Arwen, who, if you've seen the Lord of the Rings movies, is the wife of Aragorn by the end of the movie. She's also the daughter of Elrond. So rather than inserting the SIRT6 longevity gene in these individuals who chose to be counted amongst men, and are therefore mortal, Mandos will likely knock out any copies of this gene the half-elves have, and this would reduce their DNA repair capabilities, so their cells would accumulate DNA damage more quickly and would therefore age more quickly. And I also believe he would knock out genes involved in the production of the telomerase, because if he knocks out genes involved in the production of telomerase, those telomeres will not be maintained properly. And if the telomeres aren't maintained properly, that will result in damage to the ends of the chromosomes and an increased rate of aging. And I believe he will insert genes like P66HC, because that will increase the levels of reactive oxygen species in these individual cells, which would likely lead to an increased amount of cell death. So essentially, what Mandos used CRISPR-Cas9 for those who chose the fate of elves, he would do the exact opposite for those who chose the fate of men, so Elros and Arwen. And why you might choose to be immortal? You might ask, why would you not want to be immortal? Well, the answer is love, because Arwen loved Aragorn in the movies and in the books, so that just goes to show that love can and probably will kill you, just as it did Arwen. And that's my presentation, and you're probably feeling like Frodo, that you've walked through Mordor up to the top of Mount Doom, and that's kind of what I feel like too. So I hope everyone's Easter is nice, and I hope you enjoyed this video and learned a little something about CRISPR-Cas9 and some genes involved in cell aging and cell longevity.